Hey, welcome to another Study With Me video where my videos are designed to be a visual, repetitive, and interactive aid for your microbiology class. So in this video, we'll be going over the movement across the membrane and sporulation. All right, first up, we have passive processes. Now, within passive processes, there is simple diffusion, facultative diffusion, and osmosis. So here I have my triggers. First up, I have simple diffusion. Now here is the membrane, the high concentration and the low concentration, and I have my arrows to show which direction the molecules or ions are going. So in the back of my triggers, I have little notes to help me remember which is which and what the information is. So with simple diffusion, it's the net movement of molecules or ions from high concentration to low concentration. Now, some of these molecules can be oxygen or carbon dioxide. Next up, I have facultative diffusion for prokaryotes. So in prokaryotes, they have integral proteins. These proteins are channels or carriers. Now the ions and large molecules are what move through these proteins. Now the name of these proteins are transporters or permeases. In facultative diffusion, energy is not expended. Molecules move from high concentration to low concentration, and you see that here, from high concentration to low concentration. Now, these proteins are non-specific, so ions and small molecules move through the permease or transporters in prokaryotes. They're non-specific proteins in prokaryotes. Okay, next, I have facultative diffusion for eukaryotes. Everything is pretty much the same except that they transfer large molecules into the cell and that their proteins are specific. They transfer sugars, simple sugars, and vitamins. So they have integral proteins that act as channels or carriers. They can transport large molecules. Their names are transporters or permeases for the proteins. They do not expend any energy, and that the molecules move from high concentration to low concentration. The molecules that go through these proteins are simple sugars and vitamins. Lastly, we have osmosis. Osmosis have aquaporins. They're proteins that move water solution through the protein. There's a net movement of solvent across the selective permeable membrane. They can also go through the membrane. There is a high concentration to low concentration of solvent movement. So we have our solvent with high concentration here moving to low concentration. So that means that the solute is high when there's a low concentration of solvent, and the solute is low when there is a high concentration of solvent. And that's osmosis. So I'm going to stop here and I'm going to test your knowledge on what I just explained and see if you can get it. So I'm going to mix these up. First up is this one. If you can tell me what this is and all the information that goes with it. We have large molecules that go through the protein, no energy is expended. Is it simple diffusion, facultative of a prokaryote or eukaryote, or is it osmosis? Well, if you said it was facultative, yes. Now we're going to go in a little bit deeper. Is it prokaryote or eukaryote? Eukaryotes. They process large molecules such as simple sugars and vitamins. They have integral proteins that act as channels and carriers. They move large molecules like the simple sugars and vitamins. The name of their proteins are transporters or permeases, and no energy is expended. So the molecules go from high concentration to low concentration. Facultative, diffusion, and eukaryotes. How about this one? Simple diffusion, facultative diffusion, or osmosis. We already have this one. So this one is a movement across the membrane from high concentration to low concentration until it reaches equilibrium. If you said simple, that's correct. Simple diffusion, a net movement of molecules or ions from high concentration to low concentration until equilibrium is reached. Now the molecules that can move across the membrane, oxygen and or carbon dioxide. How about this one? It's not simple. So we have facultative diffusion and prokaryotes, and we have osmosis. Now this one is the movement of small molecules from high concentration to low concentration. No energy is expended. There are proteins called permeases or transporters. And they're also non-specific proteins. Facultative prokaryotes. Okay, next we have osmosis. Now that you know that it's osmosis, give me all the information about osmosis. What's the name of the protein? What is the movement of the solvent? From high concentration of solvent to low concentration of solvent. So if there is a high concentration of solvent, what is the concentration of solute? Now, if there is a low concentration of solvent, what is the concentration of solute? The protein is aquaporins. There's a net movement of solvent across the semi-permeable membrane from high concentration to low concentration of solvent. So that means that when there is a high concentration of solvent, there is a low concentration of solute. When there is a low concentration of solvent, there is a high concentration of solute. This is osmosis. Okay, we're going to try this again. do something a little different. With these two, I want you to give me the name of this one and its description, and the name of this one and the description. You can pause the video now to do that really quickly. All right, now that you're back, do the same thing for these two. Give me the name and the description. All right. Hope you did well on that. Next up, it's solutions. Isotonic, hypertonic, and hypotonic. So here I have isotonic. Iso meaning same or equal. Again, on these I have the information. So here, iso meaning equal. Now there is no net movement. Everything is equal. Iso equal. All right. Next one. Looks, there's a high concentration of solute or molecules inside the cell, and there's a low concentration of solute in the environment. If there is a low concentration in the environment, that means that it's hypo. Hypo meaning under or below or less. So I'm going to put less. Now, hypotonic solutions. Water moves into the cell. Now, within hypotonic solutions, there can be osmotic lysis. This is the damage or a weak cell wall of the cell when it swells. Now remember, hypo meaning less, that there is less molecules in the environment. Water goes into the cell where there's more molecules inside the cell. Lastly, hyper. Let's choose a different color. Hyper meaning more or above. And as you see in this picture, that there are more molecules, if I can pick it up, more molecules in the environment than it is in the cell. So water is going to leave the cell. Hypertonic solutions. Water moves out of the cell. In hypertonic solutions, there is plasmolysis, the shrinkage of the cytoplasm. Now remember, hyper meaning more, that there is more molecules in the environment, which means that water is going to leave the cell and go into the environment. All right. Tell me what solution this is and give me some information about it. And this one, what is the name of this solution? Give me some information about that one. And lastly, this one. What is this one? And give me some information about it. All right, let's go over it. Here, so what we see is a low concentration on the outside, high concentration on the inside of molecules. we have water going inside of the cell. So when the environment has a low concentration, you think of less, you think of hypotonic. Here, there is no movement of water. There's like an equilibrium, equal, isotonic solution. Here you see a lot of molecules in the environment. There's water moving outside of the cell. You think of a lot of molecules, more in the environment, hypertonic. So we have water going inside of the cell. When water is going inside of the cell is going to swell. So you have osmosis here in hypotonic. Hypotonic, hypo meaning less molecules in the environment, water going inside of the cell, allowing the cell to swell, osmotic lysis. Plasmolysis is in hypertonic, meaning more. So water's more in the environment. So water's leaving the cell, it's shrinking the cell, that leads to plasmolysis. And lastly, we have isotonic solution. All right, hope you did well. I'm going to do one more test. Make sure you get this. Name that one and all the information that goes with it. Name this one and all the information that goes with it. And name this one and all the information that goes with it. Now with this group, we have isotonic, hypotonic, and hypertonic movement across the plasma membrane. We're going to recap. Here in passive process, we have simple diffusion, facultative diffusion, and osmosis. Now, active process is another way that molecules and ions move across the plasma membrane. Now, in active process, the protein is called transmembrane protein, specifically ATPases. Now, the movement requires ATP, and it goes from low concentration to high concentration. Now, since it's going from low concentration to high concentration, of course, energy is needed. And ATPases help move the molecules. Now, some of these molecules are glucose, ions, and amino acids. Now, we also have antiporters and symporters. These are proteins that move molecules across the membrane. Now, antiporters move molecules in one direction, and also move it in the opposite direction. Here's our protein. Symporters, on the other hand, move two molecules or move in two directions in the same direction. Sim meaning same, anti against. We have our protein. So antiporters move molecules across the membrane in one direction and in the opposite direction. Symporters move molecules in the same direction, two directions. Sporulation is the formation of spores. So step 1 in sporulation, we have spore septum. It isolates the new DNA and cytoplasm. 2, the membrane surrounds the new DNA and the cytoplasm. 3, the spore septum surrounds the DNA. This equals a 4-spore. So this is the 4-spore. It's 2 membranes around the newly duplicated DNA. 4, the cell wall forms between the double layer membrane. And that's here. Here, the spore is coated with a protein. This coat is called a spore coat. And lastly, the endospore is freed. All right, I'm going to mix these up and see if you can put them in order. Now, each picture is next to a number. Your task is to put the formation of a spore in order. So you can pause the video now and take a few seconds to do that. Then I'll come back with the answer. All right. We have 5, 6, 2, 1, 3, and 4. If you have it in that correct order, then you understand the process of sporulation. But if you missed 1 or 2, we're going to practice again. All right. So I'm going to put these down and we're going to try it again. Mix them up. Okay. All right, now that you're back, we're going to put it in order. Okay, we have 2. We have the spore septum starting to create. It's starting to separate the newly DNA and the cytoplasm. We have 1. The plasma membrane is separating the new DNA and the cytoplasm. 4. A 4 spore is created, and this is a double membrane around the new DNA. 6. Here, between the double membrane, we have a cell wall that's created. 3. A spore coat is formed. And 5. The spore is free. So how did you do on this one? Good, I hope. So we're going to go over these again. Here. Spore septum isolates the DNA and cytoplasm. Number 1, spore septum. Number 2, the plasma membrane separates the new DNA and cytoplasm. So it's a membrane. Number 1 is spore septum. Number 2 is the membrane separating DNA. 3 is 4 spore. 4 spore is the double membrane forming around the DNA. So we have for number 1. So what we have for number 1 is spore septum. Number 2, the membrane separating the DNA and the cytoplasm. Number 3, 4 spore. That's a double membrane surrounding the new DNA. Next, we have the cell wall forming between that double membrane. So we have the spore septum, we have the membrane, we have 4 spore, now we have a cell wall. Next, we have a spore coat that's surrounding the spore. So number 1, we have a spore septum, membrane, 4 spore, double membrane, cell wall in between that double membrane, and a spore coat around the spore. Lastly, it's freed. So we're going to go over it again. Spore septum, a membrane that separates the DNA and the cytoplasm. You have a 4 spore, that's a double membrane around the new DNA and cytoplasm. There's a cell wall that forms within that double membrane. You have a spore coat that surrounds the new spore, and we have the spore that's freed. And that's sporulation.